All right, we're almost at the end. In this little lecture, we're going to talk about synapses, which are what's going to happen when one neuron talks to another cell. So, a synapse is going to be comprised of at least one neuron, and that neuron that is going to be sending a message to another cell is called the presynaptic cell. And then the cell that it communicates with is going to be called the postsynaptic cell. And the synapse is the place where they come together and meet. So you can see that happening right here. So this presynaptic neuron is going to be sending a message to the postsynaptic neuron. So in this case, all of the cells that are shown here are neurons, right? So you've got multiple neurons talking to this other neuron here. So you've got three other neurons all sending signals to this one postsynaptic cell. The presynaptic cell is always a neuron. That's what makes it a synapse. The postsynaptic cell can be a neuron, but it could also be another type of cell. So it could be a muscle cell, or it can be any other cell that responds to a signal from a neuron. So it's this space, it's this meeting between the pre and postsynaptic cell that makes up the synapse. And the little tiny space between the presynaptic cell and the postsynaptic cell is called the synaptic cleft. Okay, so in this case, as I mentioned, the synapse is between a neuron and another neuron. So when a neuron does receive messages from other cells, it usually receives them either on the cell body or on one of the dendrites. That's the most common. So you can see here there's also a neuron that's communicating with the axon terminals of this cell, but that's not as common. This is more common to see a neuron communicating either with the cell body or with dendrites. Okay, so synapses can transmit either electrical signals from one cell to another, which we'll talk about in just a minute, or more commonly, they're going to communicate with each other by sending chemical signals across the synaptic cleft, that very small area. Okay, so just to talk about electrical synapses, this is kind of an electron microscope view of some electrical synapses, and this is more of a cartoony view. So you might remember from back, I think early in Bio 151, we talked about gap junctions, which are cytoplasmic bridges that connect the cytoplasm of one cell to another. So within the central nervous system, so within the brain or within the spinal cord, you often have cells that are going to be connected to each other through these gap junctions. So you can have the cytoplasm from one cell just flow directly into the other cell. And if there's an electrical synapse, what that means is that if there's an action potential in cell 1, that action potential can actually just flow right directly into cell 2, right? So sodium coming in through voltage-gated sodium channels at the end of cell 1, the sodium can flow through the cytoplasmic bridges, flow through the gap junctions, and trigger the opening of voltage-gated sodium channels in the next cell. So electrical synapses, they're most commonly occurring within the central nervous system. The nice thing about electrical synapses is that they allow for very rapid cell-to-cell conduction, because all it takes is just for those few sodium ions to flow directly from one cell to the next. So this is one way that neurons can communicate with each other, but more commonly, you're going to have chemical synapses. So in a chemical synapse, you are going to have the presynaptic cell in response to an action potential getting to the end of the axon, which is called the axon terminal. What's going to happen is you're going to have that trigger a series of events that will cause the axon terminal of the presynaptic cell to release chemicals called neurotransmitters. So the neurotransmitters are going to be paracrine signals. Hopefully you remember from when we talked about the endocrine system. A paracrine signal is going to be released from a cell, in this case a neuron, travels a short distance, and then acts on receptors on a target cell. So in this case, the neurotransmitter will be released from the axon terminal of the presynaptic cell, travels across the synaptic cleft, and binds to receptors on the postsynaptic cell. The neurotransmitter release is going to get triggered, as I mentioned, when an action potential reaches the axon terminal. And in a second, we'll look a little bit more closely at the detailed mechanism of how that electrical signal will trigger the neurotransmitter release. And basically what's going to happen is that there are going to be voltage-gated calcium channels in the axon terminal that will open in response to the action potential reaching the end of the axon. That's going to cause calcium to flow into the cell. So calcium is more abundant in the extracellular fluid than it is in the interstitial fluid. So calcium will flow in through those voltage-gated channels. And it's the calcium influx that's going to trigger the release of neurotransmitter from vesicles in the axon terminal. So we see that happening sort of here in this picture. So here are the voltage gated calcium channels right here. And there's a couple over here as well. So here's the axon terminal, here's the synaptic cleft. So what's going to happen here is the action potential is going to travel down the axon, gets here, it's going to trigger the opening of the voltage gated calcium channels. Calcium's going to flow in, and that's going to result in these vesicles that are filled with neurotransmitter. In response to that calcium influx, they are going to then exocytose their contents into the synaptic cleft, and it's going to travel this very short distance. It's going to bind to receptors on the postsynaptic cell membrane, and it's going to bring about some change in the postsynaptic cell, and that's how a chemical synapse works. One thing I just wanted to mention about this is that I mentioned in the last lecture that the way that a neuron can modulate signal strength is by altering the frequency of action potentials, and that is actually going to be translated by having a greater neurotransmitter release in response to more frequent action potentials. So you can see that happening here. So in response to a weak stimulus, you're going to have action potentials happening, but they're not that close together. And that's going to result in a moderate amount of neurotransmitter release. And then in contrast, you can see that if you have a bigger signal causing a bigger graded potential, you're going to have more frequent action potentials, and that's going to result in more neurotransmitter release. So that's another way that we can communicate the strength of a signal, is that the more frequent the action potentials are, the more neurotransmitter is going to get released from the axon terminal of the presynaptic cell.